Okay. Good afternoon, everybody. My name is Sephokoane Makuwa. I'll be presenting to you data on draft genome sequence of Bacillus sp strain MHSD28, a bacterial endophyte isolated from Dicoma anomala, which is a medicinal plant. The paper is written by myself and my supervisor, Dr. Sirapa Dlamini. Plant growth promoting bacteria are known to suppress plant diseases and stimulate plant growth. And most of these bacterial strains were utilized that include the species of the genera Bacillus. So Bacillus sp strain MHSD28 is a bacterial endophyte associated with the leaves of Dicoma anomala medicinal plant, which is known to have pharmacological activities such as antibacterial, anti-inflammatory, to name a few. This Bacillus is a genus that belongs to the phylum Firmicutes of the gram-positive rods. The Bacillus species are known to promote plant growth using different mechanisms such as biofertilization, phytostimulation, and biocontrol. And this Bacillus species are known as spore formers, are known to be spore formers, and thus add advantage to this a species to survive unfavorable conditions. The experimental design and materials and method that was followed in this study is that the MHSD strain was isolated. The bacterial isolation was done using the surface sterilization technique, followed by the DNA extraction using the Zymo-rich fungal bacterial DNA mini prep kit. The sequence was then sent for sequencing using Illumina MiSeq instrument, the genome sequencing as well as the de novo assembly, including and followed by the annotation was done on the Galaxy Web platform. And later classified using phylogenomic classification using the type strain genome server and OAT. The resultant draft genome of the strain MHD28 has 5.5 million base pairs with a GC content of 35.23, which falls within the range of Bacillus cereus and Bacillus related species. And this, the strain MHSD28 has a total of 572 genes, showed on the table above on the previous slide there. The number of genes are associated with plant growth promotion that include the IAA synthesis, the cytopho, stress response, as well as nutrients utilization such as magnesium, phosphate, potassium, as well as nitrogen. The strain was phylogenomically classified using a TYG strain server, which indicated that the strain has a monophyletic relationship with closely related Bacillus species, as shown on this diagram and on this tree as well. And the strain MHSD28 was further phylogenomically classified using the OAT software, which indicated that this strain is has an ortho-ANI value below the 95 and 96 with the closely related species. And this phylogenomic classification distinguish MHSD28 from its closely related neighbors and represent a prospective novel species of the Bacillus. In conclusion, this potential new species is now in a process of being described using genomic data supported with phenotypic and phylogenetic properties. And a subsequent analysis of the genome content of this strain will help identify genes important for bacterial endophyte lifestyle and also improve data analysis in genomics for studies of plants associated with the Bacillus species. Furthermore, this genome analysis of this strain will provide information to distinguish the difference between strains within the genus Bacillus at the gene level. That brings us to the end of my presentation. Thank you.
Thank you for a really interesting talk. I really enjoyed it. Could we have any comments or questions in the chat if anyone has any? We've got a question from Peter. He's asking, how was PGAP run? Is this pipeline available in Galaxy?
Yes, we first submitted our sequence to NCBI, and then they sent us a file with the genes on that file.
Okay, so that was done on NCBI side. Very cool.
Yes.
I have a follow-up question. So you've used Galaxy. What is Galaxy like? Because I don't use it myself. What is Galaxy like? Do you like drag and drop programs you want to use, or do you just select them from a list? And then once you've put your pipeline together, can you share it?
Like, you mean when you share it, you can share it with...
So say someone else wanted to try do some annotation or assemblies like you did.
Oh, okay. It's some sort of an app where you log in, then you add in your details. Then from there, your information that you added for your sequences, it will only be seen by you. The only time where you can share them is when you download the data, and then you can share it with maybe on a publication. Then that's how you can share it.
Okay.
But then yes.